So my talk will be, I'll title it today, Signaling Dower, How Instructions in the Blood Change Mitochondrial Function and Cause Symptoms of ME/CFS. And try to put this in context with, other research that's being done. So let's see. First is, what is Dower? It's a hypometabolic state, survival states that permit survival in the face of life-threatening environmental conditions. It comes from the German word Dauer, meaning to persist, to last, or to endure. It's been studied by biologists for 50 years. And it actually led to the discovery of the first longevity genes. And what were the lessons from the genetic analysis of Dauer? Is that survival comes at a cost, and survival requires a multi-systems coordination that includes downregulation of mitochondrial metabolism, alteration of fat and amino acid, carbohydrate, nucleotide metabolism. So basically making economic use of the primary elements of life of carbon, nitrogen, oxygen, sulfur, and phosphate. And then there's a decrease in movement and decreased functional capacity. There's a hypersensitivity to chemical changes in the environment, and the animals are prone to repetitive and OCD-like behavior. And the genes show links. So the lesson between looking at Dauer from 20,000 feet is that mitochondria, immunity, growth factor, and nutrient sensing, steroid hormones, reproduction, and stress resistance all constitute a coordinated polygenic response to environmental stress that we've called the cell danger response. Now, this is a complicated table, but I'd like to just point out that there are about 15 different genes that were discovered that relate to longevity in studying Dauer. And they all have interesting mammalian homologs that range from nutrient sensing, IGF-1 receptors, PI kinase and AKT and calcium signaling, FOXO3 expression that relates to DNA repair, autophagy, and mitophagy. So these are words that are going to keep coming up because work on gene expression analysis in ME/CFS is also finding changes in these very same pathways. So TGF-beta receptor, vitamin D receptor, liver X and farnesoid X receptors for bile acids, PPR gamma, NRF2 is a frequent favorite because it regulates antioxidant defenses in cells, et cetera. So getting down to also mitochondrial respiration. So this table is just a reminder that this one phenomenon of this hypometabolic response to environmental threat involved all these different pathways. So they're coordinated. It's not just one. So this leads to our hypothesis that a proposed cause for ME/CFS is that it's a human Dauer syndrome that is triggered by the cell danger response and ultimately maintained by hypersensitivity to this ancient danger signal, extracellular ATP. And then that brings up the possibility of a treatment directed at ATP signaling. Sermon's one drug, but it is a tricky one to use, and it's never been tested in ME/CFS, and new anticholinergic drugs are needed. So our work in ME/CFS goes back to a few years before this PNAS paper in 2016, where we actually started looking at the metabolic features of chronic fatigue syndrome. And when the paper came out, a cartoonist picked this up and basically called NIH out to say, let's get going. But ultimately, the paper described this telltale signature in the blood. And what we've learned over the last 10 years since that is that the signatures of ME/CFS in the blood are actually instructions. So that's really key. The signatures are instructions. They tell cells and mitochondria to conserve energy by activating a multi-system response to environmental threat that is similar to the physiologic state of Dower. And these symptoms are maintained by hypersensitivity to ATP signaling. So our now to take a step back, I wanted to share with you a kind of our perspective about complex disease. And it started really, with the kind of training that all scientists receive, is that we really try to build deep silos of knowledge that create show what visible differences a particular disorder has from any other particular disorder, and that's an important thing and has to continue. But we wanted to look at something that I was that appeared to me to be something shared by all these different disorders, and that is that the fact that they're chronic and don't recover spontaneously very easily. And so we wanted to look at these hidden connections, and ultimately, what we began to recognize is that the process of recovery and restoration or resolution itself is abnormal in all chronic illness. So I'm going to talk now although we've looked at primary mitochondrial disease, autism, ME/CFS, Gulf War, Lyme, PANDAS, suicidal ideation, et cetera, all these different things. And by studying what was similar in those different things, we focused our attention on this process of restoration after injury that we call salugenesis. So pathogenesis asks the question, what is the cause of disease? Salugenesis asks the question, what is the cause of healing and recovery? So normally we start with a condition of health, and then there's a perturbation that can be trauma, pollution, infections, autoimmunity, microbiome, a variety of different things that leads to a state of injury. Okay, that's the process of pathogenesis. Okay, so it turns out that, and there are literally thousands of different ways that injuries can occur. But there's just one path back to health, and that turns out to be a polygenically evolved three-step pathway that has a beginning, middle, and end. It starts with a mitochondrial metabolism that changes to support inflammation, and it has to move to the activation of stem cells that restore the loss of the cells that were lost in the original injury. And then those cells that were newly born have to differentiate and change their gene expression profile to become good citizens of that tissue, whether that tissue is muscle or liver or kidney or brain or heart. It's always the same. So this process of salugenesis is the same whether it's initiated by a paper cut, a scraped knee, or a pneumonia. Okay. Interestingly, it turns out that along this process, exposures to a variety of different environmental toxicants or re-injury itself or reinfection can inhibit progress through this process. And when that happens, more of this primordial danger signal of extracellular ATP is released, and that in turn inhibits progress along the way. And when this process is incomplete, it results in accelerated features of aging, and that's for another talk. But mainly I want to just point out that there's a difference between treatments that are directed at the original pathogen or pathogenic process, and the treatments that are directed to try to unblock the steps of healing, called salugenesis. And so why don't we have any salugenesis therapies yet? And I believe it's just because we didn't know how to look, but that's changing. And so, and Yeah, I'll leave it at that. So here's a way of looking at our healthy circadian rhythm. We have a period of wakeful activity, nutrient intake. There's waste and toxin removal, social connection, nature connection, and restorative sleep. With activities, cells release a normal amount of extracellular ATP that builds up, is converted to adenosine, which helps initiate sleep. Ultimately, that is metabolized, and we wake up and start the day again. It turns out that under stresses or pathogens or other triggers, that pathogenic process leads to an acute illness that includes an increase in extracellular ATP. That will also cause this phenotypic conversion between pro-inflammatory M2 mitochondria the anti-inflammatory M2 to pro-inflammatory M1 mitochondria, and you can see a structural change associated with that. Well, that actually is the beginning of this CDR1 phase, or cell danger response 1 phase of inflammation that uses glycolytic energy metabolism. That transitions to the CDR2 phase that uses aerobic glycolysis, which is Warburg metabolism, which you may have heard is important for rapid cell growth and is used by cancer cells, in addition to the intestinal epithelial cells and bone marrow cells. And then there's this CDR3 phase, which is a restoration of mitochondrial oxidative phosphorylation and M2 anti-inflammatory polarized mitochondria. But they're off ramps, and one of these is this hypometabolic survival state that we believe is what people with ME/CFS symptoms are cycling in. And with a variety of stressors, it'll cause this shift back to a more glycolytic metabolism that we believe is part of the post-exertional malaise phenomenon. And this oxidative metabolism, I will point out, is actually something that is something. It's something you would predict when ACOD1 is activated and itaconate is produced. Again. The whole process of health and healing together are the process of recovery or of healing, of salugenesis. So what is the cell danger response? It's governed by over 5,000 genes and chronically by up to 40 different key metabolic pathways. It's a universal response to environmental threats. And so really healing cannot occur without it. CDR begins with mitochondria in the cell and spreads in a signaling cascade to neighboring cells, the vagus nerve, across anatomical scales and space and time. It's biologically expensive. It is energy and resource consuming, and until it's resolved, athletes and soldiers will underperform, aging will be accelerated, and the symptoms of chronic disease and disability will persist. So the acute CDR is something that's necessary and normal for the completion of healing. But the chronic CDR is abnormal and produces symptoms of chronic illness. So just to put this, to underscore this, is that from the point of view of a cell, when ATP is inside the cell, that's a good thing. When ATP is released from the cell, it is releasing the most expensive thing that the cell makes. And as a consequence, it is a primordial danger signal. But ATP can be metabolized to a variety of different things, down to adenosine, to inosine, to xanthine. And xanthine itself has behavioral effects. It affects T cell development and a variety of different things. Just to show you how all life forms on the planet evolved to be able to use this cell danger response that's coupled to initiated by ATP, but ultimately coupled to intracellular calcium. Here's a little mustard plant, a Arabidopsis, that's been engineered to fluoresce when calcium is released inside its cells. And when you just add ATP to this little cricket bite over on one side of the cell, in less than a minute and a half, that danger is communicated to the rest of the plant. And likewise, you can take a dish of microglial cells, these inflammatory cells in the brain, and whoops. Let's see. Oh yeah. So and you can put a drop of ATP in the plate, and then ask what happens. And it turns out the microglia become activated, and they send their pseudopodia, to surround the signaled danger. So ATP is a key regulator of the cell danger response. Here's a cartoon of a cell, and ATP is released through channels in the membrane that open and close in proportion to the amount of stress the cell is under. And the mitochondria, of course, are communicating to the nucleus. They're sending metabolites that are used to regulate epigenetic modifications of histones and of DNA in the nucleus. When the channels open up, that results in loss of intracellular ATP to extracellular ATP. So we went looking and wanted to find out, okay, are there drugs that can actually reduce the loss of that ATP and inhibit the binding, that signaling of danger through those P2X and P2Y purinergic receptors? And Sermon was the only drug available. Hopefully there'll be other drugs soon. So ATP looks like this. It's a molecule that, like many in metabolism, that has dual functions depending on where it's located. For ATP, if it's located inside the cell, it's matter and energy. If it's outside the cell, it's information. So ATP, interestingly, is more universal than any gene on the planet. It's inside the cell, as I said, it's matter, outside, it's information. And so the receptors, there are 19 different purinergic receptors. They go by names of P2Y, P2X, and P1, which are the adenosine receptors. And they're actually more widely expressed than any neurotransmitter or cytokine receptor. So more than dopamine, serotonin, GABA, et cetera. Okay. So ultimately, what we see is cells that survive the initial infection or stress or trauma. Ultimately, if that trauma is not resolved, or that the healing cycle is not completed, the surviving cells become hypersensitive to ATP signaling and maintain this chronic CDR. That's true for all these different disorders, but we really focus on ME/CFS. We use metabolomics to identify some of these metabolites, and it's not a simple story. I mean, there are a lot of things that are co-regulated. And there's a network control architecture that's we think traces back to a root cause of this purinergic signaling that regulates all the other things. It's a reminder too, though, that really all the good science that's being done is true. It's just a layer of the problem. And ultimately, to help patients, we have to find the layer that is most amenable to intervention. And okay. So when a cell is stressed, it releases ATP. And because of the overlapping expression of different purinergic receptor sets on specialized cell types, different cell types will respond in specialized ways. So for example, when mast cells are exposed to ATP, they'll release histamine and tryptase and all the other things that we know mast cells do. When microglia are exposed, they'll release IL-6, IL-1 beta, and they increase their extracellular vesicle release. Cortical neurons, calcium will be released. Also, extracellular vesicles, there'll be a lowering of the seizure threshold. Neutrophils also express purinergic receptors and actually will use ATP to chemotax to a site of injury. And it'll And ATP production will stimulate NADPH oxidase or NOx activation for reactive oxygen production. The monocytes and macrophages are particularly, active specialist cells in this early stages of the cell danger response, and they will produce a whole host of different things uh, in response to ATP signaling. And then finally, when you get to muscle cells, ATP signaling will actually inhibit oxidative phosphorylation, so that oxidative production of ATP, and it'll recruit macrophages and T cells into the muscle. All right. So this slide, I just summarized this emerging evidence from the last decade, which has been that when you do adoptive transfer experiments, where you take the plasma of a patient with ME/CFS and use it to treat healthy cells in a variety of different ways, it causes that plasma and contains the instructions that change the mitochondrial and cellular function. So Ron Davis and his nanoneedle work actually showed that not only do the PBMCs of patients with ME/CFS have this interesting response to stress, an impedance response, but if you take the plasma of patients with ME/CFS and use it to expose to healthy PBMCs, their stress response will be altered. Likewise, red cell deformability or stiffness is influenced when you treat normal red cells with ME/CFS plasma, in addition to the fact that red cells isolated from patients with ME/CFS also show this increased stiffness. Bhupesh Prusti and I published a paper on how you could take plasma from a patient and use it to treat cells in a dish and cause mitochondrial fragmentation and an interesting antiviral effect. And then we have a paper in review right now that is showing that immune complexes can transfer some of that mitochondrial fragmentation and hypometabolism phenotype. And the newest of all is some work that we're doing with the plasma from patients that were in that original 2016 PNAS metabolomic study that we published, and using the ME/CFS plasma and control plasma to treat muscle cells, the C2, C12 cells, in a dish and look at how mitochondria respond. And what happens is that mitochondrial oxidative function goes down, but so does the cellular glycolytic ATP production. So both of those are decreased. All right. So I'll finish up with a couple slides. Before the COVID pandemic, the annual economic impact of ME/CFS was estimated to be $36 to $51 billion. After the pandemic, the annual estimated cost is $149 to $362 billion in healthcare costs and lost wages in the US. If CERAMENT or any treatment could help just 10% of patients recover from ME/CFS, it would add back a tenth of that total amount, or $36 billion, to the US economy. And since the NIH budget for ME/CFS in 2024 is 10 to 13 million, which is down from 15 million in 2017, the impact of that successful treatment of just 10% in just 1 year could pay for over 2,000 years of ME/CFS research at NIH. All right, so here's my scientific summary: that the mitochondrial dysfunction observed in ME/CFS patients is not actually a dysfunction. That mitochondria are doing precisely as they are instructed by signals in the blood and from the brain. They're not damaged, diseased, or in dead-end exhaustion. The mitochondria are being actively downregulated by signals that circulate in the blood and create this hypometabolic survival state that's similar to Dauer. This physiologic state can be traced back to ATP signaling. But the overall message is there's hope. Mitochondria and patients with ME/CFS can recover. and the results establish a mechanistic foundation and incentive for accelerated development of new anticholinergic drugs like Suramin to desensitize the ATP hypersensitivity. But I hasten to say that also any other target that plays a role in symptoms of ME/CFS, you know, will help patients. When we develop new drugs, will help patients. Ultimately, the goal will be to normalize the cell danger response and unblock salugenesis and permit cells to extinguish the mitochondrial dysfunction that causes the symptoms of ME/CFS. And several other chronic multi-system fatigue disorders show similar biology. So not only long COVID, post-Lyme, and post-chemo syndromes, but many others. I'll just end with this final note that I believe that salugenesis is the medical dark matter of the 21st century. We spend really all our time, thinking about pathogenesis models that affect acute injury and how that injury happens. But ultimately, what we're left with is systems that are unable to resolve inflammation, that are unable to choreograph the sequential steps necessary of changing in mitochondrial function to get us back to health. And that is the study of salugenesis. So I'll thank my financial supporters, and particularly UCSD Christini Fund, and say thank you, and I'll end there.